Hello class and welcome. This is a quick review of Unit 1. There's a lot of stuff, but we're going to go through it with a bit of pace and see how much you remember. So, the four things that we learned about in this unit are dealing with the cell cycle, asexual reproduction, cancer, and cloning. And there's a lot of information about those. So I hope this is useful. First thing, you should know the cell cycle really well. So two main stages of cell cycle, you should know that those are interphase and mitosis. Okay? And you should also be able to identify the different subphases of those stages. So interphase has three different subphases, which are G1, S, and G2, and you should know what happens in those three. And mitosis, prophase, metaphase, anaphase, telophase, cytokinesis. Okay, so you have to know them in order. It's PM, ATC, PMAT, and then C. Okay? So, interphase, subphase G1. This cell here has a nucleus with two different types of chromosomes. Okay? Chromosome number 1 from dad and chromosome number 1 from mom. Chromosome 2 from dad, chromosome 2 from mom. During S phase, they replicate. Will I ask that on the test? Yep. And then we get the second growth phase, G2, where it continues to grow and it makes more proteins that the daughter cells will need after it divides. Okay, mitosis. Within mitosis, there are 5 subphases, and prophase is the first one. And what happens? Well, we get the centrioles moving to opposite ends. You don't need to know that name, but you do need to know the rest of this. You need to know that the chromosomes go from chromatin form into X shapes. Okay, so they wind up really tight to form X shapes. You should also know that the nuclear membrane breaks down and that also the spindle fibers grow and that they attach to the chromosomes. So lots going on in prophase. You need to know that. The next one, the easiest thing in size 9, boom, metaphase. Metaphase, they meet in the middle. And if you guys all remember that on the test, I will buy you all a pony. Okay, I really hope this is the year that everyone remembers that in metaphase. They meet in the middle. Ponies. Next, anaphase. Anaphase, they separate. Okay, so the chromosomes get pulled apart to opposite ends. And then we get telophase, where there's a lot happening again. But if you can remember prophase, then you can remember telophase, because telophase is basically undoing what happened in prophase. So something's going to form, something's going to break down. There are no nuclear membranes here, so that's going to have to form. And the spindle fibers are going to break down. Okay, so spindle fibers break down, boom, they are gone. Nuclear membranes form. And then what else happened in prophase? The DNA formed these X's from chromatin. So boom, they go back to chromatin. Okay? All right. Then we get the last part, which is cytokinesis. And in cytokinesis, they are going to opposite ends. Okay? Like that. And splitting in two. There we go. Okay? 2 daughter cells. We just showed the DNA, but of course they've got all that other stuff in there. Mitochondria, Golgi body, etc. Okay? That is cell cycle. Next, asexual reproduction. So we learned 5 different types of asexual reproduction, and there's a lot to know there. You need to know the name of each type, a description of it, how it relates to the cell cycle, and an example of an organism that reproduces that way. Okay? So first type, Type 1, binary fission. Okay, what's happening is you get a single-celled organism and it replicates its DNA and splits in two. Okay, so very, very simple form of asexual reproduction. But because it's replicating its DNA here, both of the offspring have the same DNA. So they are clones of each other. And what kind of organisms reproduce in this way? Just single-celled organisms. Okay, like bacteria. How is the cell cycle involved? Well, it's basically just doing the cell cycle. It is replicating the DNA and dividing. Okay, so goats do not reproduce this way. They don't replicate their DNA and go, but bacteria, single-celled organisms, can. Type 2, budding. So this is a hydra, and so organisms like this that have only one or two cell types can undergo budding. And so they do cell division like this guy to form a bud, and then the bud breaks off, and it will undergo more cell division to produce a clone. Okay, so that is an example of asexual reproduction. These guys have the exact same DNA. They are clones of each other. So how is cell division involved? Well, it does cell division to produce a little buddy right there. And then the bud breaks off and undergoes more cell division to produce a clone. An example, a sea sponge or a hydra. Okay? Occurs in organisms with very few cell types. So ponies cannot reproduce this way because they have many cell types. They have skin and hair and muscle and bone and etc. Okay? But something like a sea sponge, one or two cell types, does cell division to produce a little outgrowth that pops off and bam. Type 3, spore formation. Okay, so spore formation, like in this puffball, which is a type of fungus, it does cell division to produce these spores. Okay, and same with ferns. Ferns can reproduce sexually, but they can also reproduce asexually. And so these clusters of cells on the bottom are spores. Okay, so how is cell division involved? Well, it does cell division to produce those tough cells called spores. And when those spores fly out, then they will undergo more cell division to produce a clone. So this spore and this fungus cell right here will have the exact same DNA. Okay? And when that spore comes out, this spore undergoes cell division and produces this one. So these will have the same DNA as well. In fact, all of these will have the same DNA. They are clones. Okay? So this is typical in plants and fungus. Okay? Type 4, fragmentation. An organism like a sea star, whoop, gets cut. And if there's enough of that central disc, these things can do cell division to produce the missing parts. Okay? And so sea stars are capable of this. There are many plants that are capable of this. We talked about Eurasian milfoil, which can get transferred from one lake to the next if it gets stuck on your propeller, and then you go to another lake and you have a little piece on your propeller, gets into the next lake, and it is happy there to start dividing and fill that lake up. Okay, so what's an example? A sea star is an example. What is happening? Well, the organism is cut and it does cell division to produce the missing parts, and so you get a clone of the parent. So we're going to get two sea stars that have the same DNA as that. Okay, so that is rare in animals, much more common in plants. Last type, vegetative reproduction. Okay, so plants can reproduce sexually. We'll get to that next unit. But they can also reproduce asexually. Okay, and these are all examples. Okay, for instance, the strawberries, they can do cell division to produce these long runners, but then all of a sudden it does cell division not to grow longer, but to produce roots and shoots. And so you get a new strawberry plant that has the same DNA as the parent. Okay, and you can see that here in this succulent as well. The leaf touches the ground, and that stimulates those cells to do cell division to produce new shoots and roots. Okay, so this is common in plants, obviously, and not in animals. So here we have a beautiful strawberry plant producing a runner, and it does cell division to produce new roots and leaves, making a clone. Okay, occurs only in some plants. All right. Next, we talked about cancer. And cancer is an important part of this unit because the thing about cancer is that it is a group of diseases where the cell cycle, cell division is out of control. Okay? And so in this section of the unit, there are 7 different terms that you need to have a good grasp of. Okay, and the first one is mutation. So mutation is a mistake in our DNA. Okay, and why is that a problem? Well, because your DNA is instructions to make protein. So if there's a mutation, a mistake in the DNA, then it can't produce that protein properly. Okay? Now, certain mutations are more harmful to others, more harmful than others. If I get a mutation in my skin cell for the gene for insulin, because all of my cells have the same DNA, that is not a problem. Okay, why? Because that cell is not making insulin. My pancreas makes insulin, but my skin cells don't. Okay, but there are other genes that code for other proteins that if those get a mutation, that can lead to cancer. So we want to avoid things as much as possible that can cause mutations. Okay, we don't want to get massive sunburns constantly. We don't want to smoke, and we want to avoid other chemicals that can cause mutations. Okay, term number 2, tumor. So what is a tumor? A tumor is simply a lump of cells dividing out of control. Okay, and why is that a problem? Well, if it's a tumor in let's say your brain or in the liver or in other tissue, it can disrupt the function of that tissue. So it could affect your brain function and it could affect your liver function. Okay, and so these are dangerous to our health. Okay, so a tumor is essentially a lump of cells dividing out of control. Term number 3, checkpoint proteins. So hopefully you remember checkpoint proteins. Okay, checkpoint proteins are proteins that are in your cells, and they basically regulate the cell cycle. They make sure the cell cycle is running properly. Okay, and so they're monitoring things in the cell, and if there's a problem, they will either try to fix the problem. Okay, God has designed cells very well, or they will cause the cell to destroy itself, which again is a very smart, wise part of the cell. Okay, part of God's creation in the cell. Because if there is a problem, if that cell becomes cancerous, then hopefully these checkpoint proteins can shut down that cell so that it doesn't develop into cancer. Okay, now getting a mutation in checkpoint proteins, we want to avoid that. Okay, that is dangerous because then the cell cycle is not regulated, and then the cell is more likely to become cancerous. Term number 4, apoptosis. The other phrase that we used for this was programmed cell death. Okay, if there's a problem with the cell, then it can undergo apoptosis and destroy itself. Okay, so apoptosis can be triggered by either the cell itself, where the cell recognizes there's something wrong and it destroys itself, or as we saw in this animation in class, white blood cells can actually detect cancerous cells, go through the body, and trigger apoptosis so that the cell destroys itself. Okay, so that's another term I'll be asking you on the test. Term number 5, contact inhibition. This one is a little bit different because contact inhibition is something that normal healthy cells do. Okay, so normal healthy cells will grow in one layer. You get a cut in your skin, and the cells will undergo cell division to heal that wound. But when they bump into each other, they signal each other to stop dividing, and you don't get a big lump of cells where you had that cut. Cancer cells, that has been turned off. So they bump into each other and they just keep dividing, making a tumor. So you should know that contact inhibition is something that healthy cells do, cancer cells do not. Okay? Term number 6, we are moving along. Angiogenesis. Okay, hopefully you can remember this one as well. Big word. The word genesis is in there, so maybe it's helpful to remember that. Okay, angiogenesis, when a tumor grows, it needs to have a blood supply. All cells need to have a blood supply. And so when it grows to a certain size, it'll actually send signals for new blood cells to grow toward it. Okay, it's the start, the beginning of new blood vessels towards that tumor. So genesis, start, beginning. Okay, genesis, you get the idea of making, creating. Okay, and so that allows the tumor to grow larger. Okay, the fact it will send signals for new blood vessels to grow toward it so that it can feed itself. If we are going to use the medicine God has given us to fight cancer, we have to treat cancer with a certain level of respect. It is from a disease perspective, a smart disease. And you can see that with angiogenesis. It is sending signals out to get new blood vessels growing toward it. And there are treatments out there that try to block that process so that it can't get new blood vessels growing toward it. Last one is metastasis. And metastasis is when cancer cells will break off a tumor and move to a new location. Okay, so some tumors won't be able to do that, but other tumors that can happen where one of the cells breaks off that tumor. Let's say there's liver cancer, it breaks off of there and starts cancer on another tissue. Okay, and so we definitely want to try to tackle the cancer, treat the cancer before it metastasizes. Okay, so that's another important term we need to understand. The last topic that we talked about was cloning. You should understand reproductive cloning and therapeutic cloning. So reproductive cloning, we are taking a cell from a person, from a guy or a girl, doesn't matter, removing the nucleus from that cell, then taking an egg cell from a woman, so only women produce egg cells, and taking its nucleus out. And egg cells and sperm cells have only one copy of each chromosome, as you can see here. But if we put this, let's say, skin cell nucleus into this egg cell like this egg cell now thinks it has been fertilized because it has two copies of each chromosome. So it's going to start dividing, forming an embryo that can be placed in the woman, allow for normal pregnancy, and produce a baby. Okay, and so we've done this with things like sheep and other organisms. We have not done this with humans, and I think we need Christians in the sciences to say, let's not do that. Because humans, while they are a gift from God, babies are a gift, we should not, start factory making them. Okay, and then we also talked about therapeutic cloning, same idea, but at the embryo stage, they're applying certain chemicals to basically turn all of these cells in that embryo into one type of tissue. Okay, so that you could perhaps heal some diseases with that. What's the problem? Well, this is human life. Okay, and so we want to respect human life as a gift from God and not monkey around with it like that, because this process aborts that baby. So the temptation to heal a disease like that, I get it, but it comes at a cost. So I hope that's helpful. I hope you feel comfortable with this unit. There's a ton of stuff in here, but I do think you've shown over the past couple weeks that you get it quite well. So have a great class.